And I'm going to mute in 1 minute after I introduce Freddy. I'm Mitchell Ho from NCI, and it's my privilege to introduce Dr. Freddy Escorcia. Freddy is a radiation oncologist, and he heads of the laboratory of molecular radiotherapy with the molecular imaging branch at the NCI, the Center for Cancer Research. And the goal of his research has been always focused on developing novel tumor-targeted imaging and therapeutic agents for pancreatic cancer and liver cancer. And he's a physician, scientist, early investigator. And the title of his talk is already on the screen. It's about targeting Glypican-3 for imaging and therapy of hepatocellular carcinoma. So Freddy, the podium is yours. I'm going to hide myself and mute myself so we don't get much noise except you. Okay, yeah.
Only the noise coming from me. Thank you.
Okay.
Introduction, and Anu for the invitation. Of course, everybody in the liver cancer program for allowing me to chat, and everybody tuning in. I know we get a lot of these meetings of late, and they can be a bit of a chore, so I appreciate you taking the time to join in. Okay, so I figured since I'm a newcomer, a bit of a bio might be in order. I did my undergraduate training in bioengineering and chemistry at the University of Illinois at Urbana-Champaign. I just learned that Mitchell, and I have a connection to U of I. You know, that sort of frames the bioengineering thought process, frames how I approach problems. And when I went to the MD PhD program in New York, I wanted to leverage that expertise, you know, develop a new targeted agent to something. I didn't know at the time that it would become cancer, but hopefully test it in vitro, validate all that, do in vivo studies, and eventually get this into human trials. Of course, doing human trials as a graduate student is very challenging, but you know that's the holy grail for the physician scientist. So I found my thesis mentor who had been working on developing targeted agents to tumors or tumor targets or tumor microenvironment targets, and leverage that to deliver radioisotopes that are cytotoxic, in particular alpha particles. And we'll review a little bit of the radiation science on that end. And you know, as a graduate student, I remember being sitting for lunch and asking David Scheinberg, my mentor, you know, what should I be when I grow up? And without pause, he said, radiation oncology. And he's a medical oncologist, so I found that curious. So, you know, when I went back to med school, I did a rotation, and sure enough, I was interested in it. The physics, the technical aspects of it, the computational components were very attractive to a nerd like me. And I completed my radiation oncology residency out there with interest in gastrointestinal malignancies, including hepatobiliary, HCC, and pancreas. So let's do a little science here. Some background on radioactive decay. So all of these types of radiation emissions are used clinically today, right? We have alpha particle decay, which emits a helium nucleus, has mass, right? And it has energy, and we can leverage that to kill cancer cells. It can be exquisitely cytotoxic. Radium-223, or otherwise known as Xofigo, is currently approved to treat patients with metastatic castrate-resistant prostate cancer with bony metastases. Radium is sort of a calcium mimetic, so it tracks to areas of bony turnover as occurs in metastatic disease. So that's currently used and approved clinically. Excuse me. Beta decay is the emission of electrons. And this has been used for decades with iodine-131 for thyroid cancers, and that's been very helpful for treating those diseases. The antimatter equivalent to the beta particle is the positron, and we use that all the time in the clinic for positron emission tomography, or PET imaging, right? With gamma emission, I'm a radiation oncologist. This is near and dear to my heart. We release gamma rays, or high energy X-rays, if you don't want to be so much of a purist. In radiation oncology, we can use actual radioactive sources. So cobalt-60 has been used historically and is currently used for things like radiosurgery. We can zap tumors in the brain, for example, with very high doses of radiation and ablate them. We can also use this with linear accelerators for radiotherapy. And this is an example of linear accelerator, right? If you'll allow me to digress a bit and share a little bit about the radiation oncology, this is a linear accelerator. This is the gantry here. The X-rays come from the head of this machine. In front of the gantry, you have these things called the multi-leaf collimators, and they move dynamically to modulate the beam. We use complex computing to be able to do this. And the end result is that we're able to generate very complex volumes of radiation deposition inside patients into deep-seated tumors. So I think that's very cool, and that's why I do radiation oncology. And the goal here, of course, is to maximize the tumor to maximize the dose rather to the tumors and avoid the dose to normal tissue. So, to illustrate what we do in RadOnc, I wanted to share a case that's not liver, but pancreas, so let's say related, just as an example. Right? So this is cross-sectional imaging of a patient with unresectable pancreatic adenocarcinoma. What you can see here, this hypodense region is the cancer. This is the tail of the pancreas. This is the duodenum right here. And these two bright spots are fiducials that we had placed endoscopically within the tumor, and that'll become relevant in a little bit. So this patient underwent FOLFIRINOX treatment given the fact that they had unresectable cancer, didn't progress, but still unresectable. What do you do under these circumstances, right? My medical oncology colleagues who might be tuning in will be the first to admit that the conventional radiation treatments that have been attempted in these types of patients don't actually do anything when it comes to survival, but they can help when it comes to local control. But that's the old type of radiation. Is there something new that we could potentially entertain and maybe help patients a little bit better? So how would we go about doing this? We delineate the normal tissue. The one that I care the most about in this case is the duodenum. We don't want to put radiation dose that's too high there because we can perforate, and that's obviously bad. The contour to the tumor is defined in red, and we contour out those gold fiducial markers to help us see the tumor using X-ray imaging. All right, this is an example of the dose wash. I'm just showing you a single slice, but this is a three-dimensional volume that we're depositing. Everything encompassed within that volume is getting what we call an ablative dose. So basically, without getting into it too much, the radiobiology of this suggests that anything getting this dose or higher that we're giving will result in us being able to control the tumor greater than 90%. So excellent local control. In addition to that very high ablative dose that we're delivering to the pancreas, we can deliver a lower dose for subclinical disease, micrometastases that we don't visualize on any of the scans. Some of that dose is getting to duodenum, but that's well within the tolerance. So we already know how much these organs can tolerate, and we stick to those constraints. All right, so this thing rotates, of course, in 360 degrees. We have those multi-leaf collimators, the MLCs, that are modulating the dose and depositing that three-dimensional dose into the patient. The one thing that I'm not mentioning is the movement of the tumor, right? And that's where those fiducials come in. So here what we're seeing is a contour. So I'm drawing out where the fiducials will lie, and these are the fiducials. And what we're seeing is a single X-ray shot as we are entering the breathing cycle that we've predefined where we want to be treating the tumor, right? So within that the breathing phase, we know that the tumor isn't moving very much, and we're able to turn on the beam because we're in the appropriate phase and the tumor is within our targeted field. All right, so normally this doesn't project very well on Teams, but I think on WebEx it usually does, so we'll try that. Okay, so we're seeing every single time we enter the gate, the breathing gate, where we define that the tumor is in the right side, we are taking an X-ray and then treating. And what you see is that every single time we take that picture, that X-ray, we see those fiducials within that volume, right? And this is one of the ways that we can help control this. You see in yellow is where we are at the appropriate gate, and that's the only times that we're treating. We're not treating the rest of the time. So beam is on, and the beam is off, the beam is back on again. And it allows us to very precisely deliver the dose where we want it to, but not where we don't, okay? The patient has pancreatic cancer. Pancreatic cancer can generate CA19-9. Carbohydrate 19-9 is a tumor marker. And you could see when the patient first came into us, he already was making, his tumor was making CA19-9, normal is 37. And it just kept rising while we were doing the workup and everything like that. We treated, and with that dose that we just went over, using that approach, and we dramatically dropped this. So we were very excited about this. We know we can deliver this ablative dose to the local disease. The issue invariably that happens, though, is that the patient can develop metastases. So when we saw this bump in the CA19-9, we looked for metastases. Unfortunately, he had lung metastases. This was preceded by several scans before we even did this radiation dose, where he had no evidence of other disease. But it goes to show you that even though our local ablation can help, it does very little when it comes to any metastases that we see. Okay. All right. So what can we do under those circumstances, right? And this is where my science sort of comes in. Right? Can we build a tumor-targeting ligand that can be cytotoxic? Right. So first, you have different components to this: a target, right? Biomolecule that recognizes that target, and then some chemistry that attaches your cytotoxic agent. For antibody drug conjugates, which you may be familiar, you attach something like a chemotherapy type of small molecule that's very cytotoxic. For us, we could do an isotope that could be used for imaging or therapy. So why do I think this is a good idea? What are examples of this thing working? So one major example and a relatively recent breakthrough is in neuroendocrine tumors. This is DOTA-TATE. It's a modified octreotate, which is taken up by neuroendocrine tumors to their serotonin receptors. And scientists modified it to attach this DOTA. DOTA is a chelating molecule that can bind radiometals. In this case, it is chelating a gallium-68, which is a positron emitter. So we can use it for PET-CT scans. Right? So what does this look like when you inject this into patients that have neuroendocrine tumors? You see these dark spots representing the neuroendocrine tumors. We have a tumor selective imaging agent that's able to localize to areas of disease. Now, what happens if you were to use the same ligand functionally and swap out the radioisotope from a diagnostic one like gallium, the positron emitter, to lutetium-177, which is a beta particle emitter? It's electrons, right? So therapeutically, what does this do? And this was studied in a phase 3 trial in Europe called the Netter trial. And what you can see here is the Kaplan-Meier, the experimental arm, had significantly better overall survival compared to controls. And this is overall survival, right? A lot of the agents that we're talking about and we're seeing in oncology these days, we're looking at overall response rates or maybe even progression-free survival. This is overall survival. So this is very impressive from that perspective. Other examples include PSMA. PSMA stands for prostate-specific membrane antigen. Okay, and one issue that we run into with prostate cancer, as well as other cancers, is that the FDG PET, so the fluorodeoxyglucose PET, isn't really taken up by prostate cancer. Here's an example here, FDG, and you see there's really nothing here. When you do a PSMA imaging agent, prostate-specific membrane antigen, which is overexpressed in prostate cancers, you start seeing uptake in this otherwise occult lesion. You can also see some non-specific uptake in the salivary glands, which becomes important when it comes to toxicity. So what happens if you couple a PSMA targeting ligand to actinium-225? Actinium-225 is an alpha particle emitter. Again, it's a helium nucleus, a lot of mass, a lot of energy deposited over a short path length. What does this do therapeutically for patients? And here you can see a patient that has a PET scan, a PSMA PET scan, and you can see there's a lot of disease in the axial skeleton. And after 3 rounds of actinium-labeled PSMA targeting ligand, you can see a dramatic radiographic response. And then with another cycle, you see a complete response when it comes to biochemical response with PSA. PSA is produced by prostate cancer. All right, so very promising, I think. So what would happen, if we try to apply this to our patient, right, with the pancreatic cancer, we needed a target. And during my postdoc work at Sloan Kettering, we identified MET as being overexpressed in several patient data sets. MET is a HGF receptor, and MET itself is a receptor tyrosine kinase involved in cell growth, motility, proliferation, among other things. Right? So here's patient data sets. Some of these are microarrays, where you can see in normal tissue in blue, lower expression compared to tumor, and this is 4 different data sets up here. When you look at the TCGA data set, comparing high expression of MET versus low, high expressions associated with decreased overall survival in patients with pancreatic cancer. When you take tumors from patients and stain them for MET, this all sort of lines up. Normal pancreas, not so much MET staining, but when you do stain, you get positive staining. So this seemed like a reasonable target for us. What happens if we were to develop a MET targeting ligand and couple it with a positron emitter? In this case, we used an antibody called onartuzumab. It's a single arm antibody that was developed by Genentech. We coupled that with zirconium and did some animal imaging to see what this would look like. We did this across multiple cancer lines, including BXPC3, one of the common pancreatic cancer lines. And what you're looking at here is a CT scan. Here's the head of the mouse. Here's the subcutaneous tumor in coronal and axial sections. And you can see we have good uptake and persistence up to 120 hours. Right? So good imaging. Does that translate into it being a decent target for therapy? So we took the zirconium out, the positron emitter. We brought in the lutetium-177, the beta emitter, and we looked at therapy. So control saline, and then just the naked antibody unlabeled, and then the late radiolabeled isotope antibody, lutetium antibody. And you could see there was a significant local control effect on the tumor growth delay and good overall survival. Right. So, I think that this is an option potentially for this. Whether this is the appropriate target in the context of a real patient, that remains to be seen. So upon my arrival here to the NCI in 2017, I wanted to see if I could apply these interests to targeted radiopharmaceuticals for imaging and therapy, and then bring in my radiation oncology interest in GI malignancies. And I learned about the liver cancer program from Drs. Gretton and Wang, which, as everybody I'm sure here knows, aims to bring together scientists and clinicians to try to address some of the incidents, the increasing incidence in mortality of liver cancer as a whole. So these are some of the folks that I've worked with personally, and I found this to be a good opportunity, given that the disease doesn't really have a tumor-selective imaging agent per se. Local radiation therapy from radiation oncology side is one of the standard options, including the stereotactic ablative radiotherapy, similar to what we did in that pancreatic cancer case that I showed you. And radiopharmaceuticals are already used, right, when in the context of transarterial radioembolization, right? So that all kind of matched up nicely. So what's our wish list, right, when we're trying to develop a tumor targeting agent? I'm thinking about it from the imaging side. We want good target specificity, target engagement to be relatively quick, good tumor penetration, and rapid clearance. If the clearance is too slow, the blood pool signal is too high, and our signal to noise is not very good, right? What about therapy? We obviously want this thing to be cytotoxic, and we have favorable excretion. We don't want this thing sticking around for too long in the liver, spleen, or kidneys, for example, right? So we ideally would be able to precisely control all these aspects of delivery, uptake, and excretion. These, again, are the major components of the ligands, right? We have target specificity, the biomolecule, the chemistry required to link the biomolecule to our radio isotope, and then which radioisotope would we be using, right? So can we do this for HCC? We first need a target, right? And collaborating with CCBR, Maggie Can's group, who I learned about through Xinwei Wang, we had them look at differentially expressed genes in the TCGA and the Tiger-LC data sets. In particular, we had them funnel those differentially expressed genes through cell membranes, specific markers, right? Because if we're using antibodies or other targeting ligands, we can only target the things on the surface. Things that are intracellular are harder for us to see. So immediately we notice that GPC3 is among the highest expressed genes. GPC3, of course, is Glypican-3 that is an important marker and used by our pathologists to help define HCC. It generally participates in cellular proliferation, motility, survival, all these things that may contribute to oncogenesis. So has anybody worked on this? GPC3, of course, right? It's a validated HCC selective target. Here's an example of a phase 2 study using GPC3 as a peptide vaccine. Here's another study out of Dr. Abu Alpha at Sloan Kettering, phase 2 placebo-controlled trial with an antibody specific for GPC3. Here's a preclinical study from the group of Shu Gai who developed that same full-length antibody, in this case, a bispecific antibody, one component targeting GPC3, the other component CD3, the idea being that you bring the T cells in close proximity to cells that express GPC3 and hopefully stimulate that anti-tumor response that way. And of course, our very own Dr. Ho has developed a nanobody, a single-domain antibody, coupled with an immune toxin, and it's demonstrated some very good efficacy in preclinical studies. Right? Here's another group out of Baylor that developed CAR T cells, armored CAR T cells, to GPC3 in children that have liver tumor. So it turns out that hepatoblastoma, which is commonly seen in children, also expresses GPC3. And not to be outdone, our very own Dr. Gretton and Mitchell Ho are trying to open up a trial using CAR T cells to GPC3 in patients with HCC. I reached out to Dr. Kleiner, who I knew had a tissue microarray, and I asked him, to what extent do tumors express this GPC3? He had several samples, and about 75% of HCC expressed GPC3 to some extent. Right? So, we thought that this was a pretty good target. What about a biomolecule that can recognize this target? There are various scaffold types that we can use here. I'm showing you the full-length antibody, some antibody derivatives, including SCFEs and nanobodies. There's peptides and potentially some small molecules. Of course, the molecular weight changes, and that changes some of the pharmacokinetics of the ligand that we ultimately make. I come from the antibody world, and antibodies are, in fact, the gold standard. So I wanted to start there and see just how much uptick we could actually see in tumors before trying some of these other agents. So we now need a cargo, right? We have the biomolecule and the target, and in this case, because it's an antibody, and the blood half-life of the antibody and the physical half-life of the isotope sort of need to be matched up, we decide to go with zirconium-80, okay? Another positron emitter. So take the antibody and run it through our workflow. And the workflow here involves confirming the native binding affinity of the ligand that we start with, in this case, the full-length antibody, using cell-free and isogenic cell line assays, optimizing the bioconjugate chemistry, characterizing the conjugate. One of the things we have to be careful is how many modifications we make to the antibody and make sure that we ensure the immunoreactivity. We didn't ruin the ability for it to recognize its target. We also want to optimize the radiolabeling. Does it work? How stable is this? Is it stable under serum conditions? And then, of course, do the in vivo biodistribution. Where does it go? Obviously, we want it to go into tumors, but we also want to see where it goes not specifically. And then the pharmacokinetics. How long does this survive in the blood? All right, so here's an example of that workflow. We're using bilayer interferometry to estimate the K on and the K off. That gives us an estimate of the binding affinity, or KD. Using cell-free and cell-based assays, we can estimate an IC50, or using a saturation study with cells, we can also estimate another KD. In general, we're in the single-digit nanomolar range, and that's plenty good for our purposes when it comes to the imaging that we might want to be doing. All right, so we couple this with zirconium. We see how stable is this in serum. So we incubate this in serum at 37 degrees and then measure the stability of the radioconjugate. And up to 344 hours, when we called it quits, it was about 90% stable. Unsurprisingly, in PBS, it's not as stable. A lot of this is due to what we call radiolysis. So these isotopes are emitting, positrons, and that can obviously cause damage to the protein. So we do see some degradation over time. When we look at the blood half-life, it's about 8 hours. Okay, so We like to confirm that the antibodies obviously bind to their target. We showed you earlier using the unmodified and then the modified with the DFO, which is the chelate that can bind zirconium. We also wanted to have a fluorescent version of this to confirm binding in cells. And here what we have is a flow cytometry study using various cell lines. G1 is an engineered A431 engineered to overexpress GPC3. HepG2, 3B, and Hus7s are liver cancer cell lines with decreasing levels of expression of GPC3. And you could see that in evidenced by the flow cytometry histogram, decreasing the extent to which it shifts to the right as we go from left to right. Okay? And this is using a commercially available antibody. And then here we're using GC33, which is the antibody that Chugai, the Japanese company, developed and actually tried in phase 2 clinical trial. So here we see a shift to the right, and that shift decreases as we go from left to right, corresponding with the extent that we have the GPC3 expression. If we inoculate animals with these cells, harvest those tumors, put them on slides and stain it, we can see a corresponding decrease in GPC3 expression as we go from left to right. The fact that we see expression corresponding to the in vitro side to the tumor side is very reassuring for any future studies that we do, and often doesn't, maybe not often, but sometimes does not happen as linearly as we would like, as it did in this case. Okay, so what does this look like now? We've created the agent, we know it's relatively stable, it's able to bind its target after we do the modification. What happens when we introduce this into animals? And here we have a PET CT. Here's the head of the mouse, here's the spine, this is the tail, that's the tumor. We see very good specific uptake into the tumors, and that uptake decreases as we go from left to right, corresponding to the decrease in GPC3 expression. If we change the threshold for the Hep3B and Hus7s, you can start to make out a little of the tumor, still very low, but the G2s and the G1s were the best expressors, unsurprisingly, based on what we know about the copy numbers. We can quantify this, and we can see what we call the tumor percent ID per gram, percent of the injected ligand that we have in there within the tumor, and it is very good, 25-30% ID per gram for the engineered line, 15% or so for some of the others, decreasing correspondingly based on the expression of GPC3. When we take that same radioligand and instead of doing, say, immunofluorescence on tissue, we can do what I call immunoradiography. Just basically, it's the same functional assay, but our readout is radioactive instead of fluorescence. We can test to see if in patient tissues our ligand can actually bind. Right? We have a negative control in A431s or positive control in G1. Nice dark staining by radioactivity, suggesting that there's a lot of GPC3 expression, unsurprisingly, and very little in A431. In the patient tissue, we have somewhere in between, and that's to be expected. Again, reassuring. This is great, right? We can localize tumors. We can do this in vivo. The radioconjugate can bind to human samples. What do we do next? It's great to see it. Can we do therapy with this? And I come from a background of using alpha particle emitters and beta particle emitters. So why would we want to attach these things to the antibodies? And the prime example of why comes from the results from this phase 2 trial, using the same antibody that we showed our immunopet with, right? Here it was a placebo-controlled trial, phase 2, randomized, with cadutuzumab, that's another name for GC33, full-length, humanized anti-GPC3 antibody, and we saw no effect on progression-free survival or overall survival, right? And to me, somebody that works with antibodies and modified antibodies for therapy, what this means is that the putative cell kill mechanism of antibody-dependent cellular cytotoxicity was insufficient to have an effect in these patients. So what if we were able to attach something cytotoxic that might be able to affect the chain? Right? And mentioned before that antibody drug conjugates are pretty popular these days, and that's built on the same premise. But in our case, we use radioisotopes. So what we're seeing here is a uranium ore, and in a cloud chamber, so it allows us to see the emissions. And we see the short ones are alpha particles, the longer ones are beta particles. Okay. And if we zoom in a little bit further, we can see a little bit of the difference and maybe surmise why we would see increased cytotoxicity with the alpha particles versus, say, the beta. So the alpha is here, it's very bright, it's interacting with matter significantly, doesn't travel very far, and that combination of high energy deposited over a short path length corresponds to a high linear energy transfer, where it's depositing that energy over a short distance and can be exquisitely cytotoxic, particularly if it goes through the cell nucleus. Electrons, on the other hand, you can see they interact with matter in this cloud chamber much less so. It's wispy, they travel much farther. Okay, so given my prior experience with alpha particles, I wanted to see if we can couple that GC33, the anti-GPC3 antibody, to an alpha particle emitter and see if we can have some anti-tumor effect. One thing that I haven't introduced you to yet is the challenges of chelation of actinium, one of the alpha particle emitters, with conventional chelates. So DOTA is a common chelate used, and it's the standard of care, the gold standard for chelating actinium. This is what I did during grad school, right? And in fact, DOTA was the same chelate that was used for the dotatate for neuroendocrine tumors that's already been approved. So one of the challenges with DOTA and actinium is that you have to heat up the DOTA incubated with the actinium to 80 degrees centigrade. And when you do that, you start risking the denaturation of the antibody, right? So we were looking to see if there are other options, and until recently there weren't. But our colleague, Justin Wilson at Cornell, published a paper a couple of years back demonstrating that this heterocycle macropop, as he calls it, is able to stably chelate actinium at room temperature. So we established a collaboration with him and try to see if we can try to create the radioconjugate and test it. So we did that, and we had animals that were incubated with HepG2 tumors, and what we see here is spider plots in the sort of light gray to show you the individual tumor sizes. In the solid line, we have the mean, the average tumor volume, and the dotted lines are the 95% confidence interval, just to give us an idea of what the trend happened to be. So with just the mass equivalent antibody, we're injecting the same amount of mass that we're giving of the radioconjugate, we see very little anti-tumor effect. When we give an IgG1, so an isotype control, this is just an antibody that should not be specific to anything in our system, but just has the radio label. We know that any radio-labeled antibody will have some anti-tumor effects by virtue of being in the blood pool, and that non-specific accumulation in tumor sometimes is a function of leaky vasculature just because of the model system. And you can actually see that the slope of the tumor growth is dampened somewhat when compared to the non-specific, or rather the mass equivalent treated group. When we do the specific antibody that's labeled with the actinium, we see a further dampening, suggesting that there is some anti-tumor local control effect. Unfortunately, when we do survival, even though we see a significant difference between the IgG versus the specific antibody, this isn't as compelling as we would like. If you recall the prior study we did with pancreatic cancer model, we saw that the experimental arm was almost straight, where the majority of animals survived. Here, we saw a lot of them end up having to be sacrificed, and that wasn't necessarily because of tumor growth, where we weren't binding, but rather because of toxicity. Okay, and one of the things we looked at here, we did a CBC, a complete blood count, and also a basic metabolic panel in these animals prior to and then during treatment to see what parameters were altered during the treatment. And immediately what you could see, and this is not surprising, is that any animal groups that got radioconjugate, we saw a drop off in the white blood cell count. These are very radiosensitive cells. They're floating around in the blood and the bloodstream, and the antibody is there, so it's been ablated. And what we see is a drop off, and then it took 40 days or so for the animals to recover. That may be some reason why we end up seeing some of the radiotoxicity. When we look at the basic metabolic panel, we didn't see any changes in, say, creatinine or LFTs that would have suggested that we received toxicity there. But we're currently looking at the necropsy studies to see if we can identify more sort of gross toxicity in these animals that receive this. Yeah, and one of the issues that comes up here too is that probably directly labeled radionuclide therapy using full-length antibodies is not the way to go here. We know the half-life of the antibodies is very long, and that's a function that's by design, right? And because of it, you do risk seeing this type of toxicity. So there's a couple of strategies you might be able to employ in that case. You can fractionate the dose, so you give a little bit of dose multiple times. You could separate the antibody targeting component from the radiation part, where you have some way of recognizing the antibody that you inject first, and then come in with second with a rapidly cleared radioactive compound that recognizes the antibody where the antibody is gone. It's called pre-targeting. So those are some options, but some of the other options too are using maybe smaller ligands, right? The ligands that I've presented to you that have been successful have been smaller. And so revisiting the options, maybe we could look at SABs, nanobodies, peptides, or small molecules, right? And I should note that my colleagues that work in this space in molecular imaging, molecular radiotherapy, generally speaking, we're sort of limited in which molecules we can use to the things that are available, right? To the things that other scientists have been able to identify, or companies have developed full-length antibodies or what have you. So that's one of the things that I'm also hoping to be able to develop, maybe some new things, and I'll hopefully share a little bit in a few slides. All right, so what can we do with these small molecular range molecules, right? So we partnered with a company that was able to do yeast display, and the benefit there is that we're using eukaryotic expression system to help us identify maybe scFvs that are specific to GPC3, smaller targeting ligand. Can we identify these and then maybe use these in vivo for our purposes? So we tasked them with generating anti-human and anti-murine sort of dual affinity ligands, scFvs, and we were able to identify some that were single-digit nanomolar specific for human, and single-digit or low double-digit binding affinity for murine. This is great. This allows us to potentially explore model systems that have an immune system that is intact. We did flow cytometry on this. The positive control was the full-length antibody developed by Shu Gai. You could see positive binding with the cell line, the isogenic cell line that expresses GPC3 compared to the negative control, the A431s. Interestingly, you see this clone GP8, which was a good binder, at least using this cell-free base assay. It also bound to the non-specific A431, suggesting that there's some non-specific interaction. So again, part of the workflow is not only the cell-free based assays, but the cell-based assays to help eliminate some of these non-specific binders. The other clones were able to bind, albeit not as well as GPC3. But we maybe give up some GPC3, human GPC3 binding affinity by virtue of also selecting for those that are able to bind to the murine. So we're trying to optimize those. In addition to that, we're looking at nanobodies. I mentioned that Dr. Mitchell Ho has identified a nanobody specific for GPC3. And when we learned about this, we established a collaboration. And this is a component of a Flex Synergy award that we got through CCR, where we were able to site-specifically modify this HN3 and demonstrate that we were able to preserve binding affinity. In this case, it was single-digit nanomolar again. We did IC50 binding when we coupled it with zirconium in our case here. And then we did a biodistribution study. And what we see here, this is, the label didn't project here, this is the G1, so this is the positive control cell line compared to the A431 negative control, doesn't have GPC3. And immediately when you look at the tumor, we have uptake around 6% compared to 1%. So that's good, but for our purposes, not yet good enough for imaging. And when we looked at the images, it was really hard to actually see good signal within the tumors. And there's a couple of reasons for that. One of them could be that we may have swung the pendulum a little bit far on the half-life component. The antibody, full-length antibody is very long, but these are very short in the order of minutes, and we're working out some of the chemistry to try to improve the blood half-life there. So stand by. Peptides represent an interesting opportunity, and several groups have already published in that space. And we were very excited to leverage that and build on the existing literature, the existing sequences, and potentially develop some multimeric versions of these peptides to improve avidity and improve binding potentially. But the unfortunate thing that we found when we did this was that when we took the top peptides that have been published, the ones with the best binding affinity, the lowest KD, we synthesized those and we characterized them using our workflow with cell-free based assays using the biolayer interferometry for K on, K off, and then also cell-based assays. All of those peptides, the two main peptides rather, were non-specific. They didn't actually stick to the target that we wanted to. So underscores the trust but verify motto that we need to have in academia, and underscores also the value of having these sort of orthogonal assays, the cell-free and the cell-based assays, and particularly the isogenic cell line-based assays, which are critical. So this represents an opportunity for us to maybe find some peptides that don't yet exist that are specific for GPC3 as well. And small molecules. This is something that actually I'm pretty excited about. We established a collaboration with Barry O'Keefe at the Natural Products Group, and came up with an assay that's pretty unique. We're leveraging the melting temperature curve that gives us an idea of how stable a particular protein happens to be. So the rationale here is that if we take GPC3, our target of interest, and have it at regular room temperature, it's structured, it's folded appropriately, and we incubate it with a dye. As we raise that temperature, the GPC3 will start to unfold and denature, exposing more surfaces. The dye non-specifically adheres to the surface, and the fluorescence increases up until the point that it becomes fully denatured, and we maximize the fluorescence. You take the the first derivative rather, you estimate the melting temperature for the native GPC3. If you add to this a natural product, for example, a molecule, and you see a shift to the right in the melting temperature from the baseline, that suggests that you've stabilized the interaction, you stabilized the GPC3 in some way. If you incubate it and the melting temperature decreases, then you've destabilized it in some way. So the destabilizers, it's unclear to what extent what that means for us, but we're keeping track of these. But for the stabilizers, we were particularly interested in those because that suggested a more specific interaction, at least as a first pass screen. Okay, so here's an example of a heat map derived from 384 well plates. Here we have GPC3 alone, so this is our baseline, and anything above when it comes to melting temperature is a stabilizer again, anything below is a destabilizer. And you can see in green, we have a few lead compounds that are stabilizing. Just this week earlier, actually, I spoke with Barry, and he was able to identify those stabilizing molecules that do not cross-react with other molecules that we know of, and we were able to identify in the order of 10 molecules that are early leads. We still have to validate, we're going to do a dose response curve, et cetera, but I'm optimistic about those results, very excited to see what pans out. Okay, so that's sort of what we have going on. Some early data since we just started here, a lot of collaborations, of course. Hopefully I've been able to convince you that radiotherapy can play a role in many cancers, right? Including HCC. We can do this ablative therapy. It's being done mostly in Europe. In the US, the uptake hasn't been as great. To some extent, this is being led by the fact that we don't have good level 1 data. We're still waiting for the NRG 11-12 study, formerly RTOG. GPC3, I think I've convinced you or shared some data suggesting that it is an attractive target for HCC, and we can use this for tumor imaging and therapy. We showed that the immunopet can be used to visualize GPC3 expressing tumors in vivo. That's been pretty convinced of that. However, I think molecular radiotherapy with a full-length antibody is still very much a challenge, and looking at potentially smaller scaffolds, including nanobodies, as Mitchell Ho has developed, might be the way to go, or other peptides or small molecules, for example. So with that, it does take a village to do a lot of this work. It ranges, of course, from the target binding or target finding things, the biochemistry, molecular biology, expression of these molecules, then the chemistry, biochemistry, and radiochemistry involved. Too many people to thank, but obviously I thank my lab and collaborators here, CCR, and within the extramural program. So with that, I want to thank you all for your attention, and I'll take any questions. Thank you.
Freddy, that was a very nice talk. I have a question regarding the imaging.
Yes.
How early do you think you can see this? Because surveillance is a big issue in HCC.
Yeah. So there's two components to that question with respect to early. One is I think the one that you're actually asking about is, can we see this in like LIRADS-3, LIRADS-4, or something like that, where we may actually be able to move the needle on the diagnosis, right? Because LIRADS, as many people might know, is one of the categorizing ways for us to assess liver lesions suspected for HCC potentially. And LIRADS-5 is definitely HCC. Anything below that is a little bit more nebulous. That's a good question. My understanding in talking to Kleiner is that you don't really see this until you have more aggressive disease. So well differentiated, probably less so. More dedifferentiated and high grade disease, you end up seeing a lot more expression of GPC3.
But so I actually meant more. You think you can long term use this in patients with cirrhosis?
To see if you can diagnose early on, right? So
yes.
Like they don't have a lesion, maybe we see something in ultrasound. Yeah.
Because the issue is with increasing fatty liver disease, you can't do ultrasound anymore.
Yeah, I haven't tested it, right? So we have to look at it specifically. I don't see why we wouldn't. If we are able to have enough perfusion where we're able to deliver a GPC3 targeted ligand and the tumor expresses GPC3, we should be able to see it. The one potential concern would be if you get non-specific accumulation of the tracer within hepatocytes, or because it's sick liver, your signal may go up and then your signal to noise is poor. But my colleague Jorge Carrasquillo at Sloan, he had done some human imaging with the same antibody, but in this case coupled with iodine instead of zirconium, and they were able to visualize those tumors. They're already diagnosed, right? So not early stage, but we could potentially extrapolate. But we would have to do that study in order to get to the definitive answer.
So, hi, Freddy. I have a general question.
Sure.
More general. So, in terms of treatment for hepatocellular carcinoma, immune checkpoint blockade has really changed the landscape of therapy. I wonder whether you have any update in terms of to combine with radiotherapy together with therapy.
That's a good thought. Atezo bevacizumab combinations now for first line for patients with HCC. And I think so. There's some evidence that external beam radiation at various doses, and that's debatable, can have an immune stimulatory effect. The rationale is that with cell kill, you may expose some neoantigens, and in the context of an immune checkpoint inhibitor, you may be able to rev up the immune system even more so against these otherwise cryptic antigens. So there's some bioplausibility for synergy between radiotherapy and immune checkpoint blockade, and it's been demonstrated in other histologies. What's interesting is that there's some emerging data on targeted molecular radiotherapy agents having similar effect. That's emerging, really I have a skeptical eye on it, mostly because if this thing is circulating, depending on the blood half-life, you may actually be irradiating the very cells that you're trying to stimulate, right? So that remains to be seen, but there's some early data on the external beam side of this.
So I mean, most of success is still on the hepatocellular carcinoma side. I agree that some sort of toxicity-induced effect that could induce a neoantigen that allow immune checkpoint inhibition works better. But in terms of immunotherapy, cholangiocarcinoma is not that a good candidate, and there's not much good treatment option for this type of tumor. What are your efforts in terms of working on cholangiocarcinoma?
Well, currently very minimal, but if we find a good surface marker for cholangio, we could easily approach it using our workflow. Develop antibodies or peptides to this target, or leverage any existing ligands, and then see how well we're able to localize this in vitro and in vivo. And if we find something reasonable, then all the more reason to try to push that forward, particularly because the options are pretty limited for cholangio.
This is Matt Young at the extramural NCI. I had a question on the Glypican-3 as being a highly modified glycoprotein. That the actual modification may change from a normal cell into a cancer cell, which may, one, affect the antibody you're developing, and two, possibly even the peptides that are targeting, the availability of the peptides. And that may have an effect on whether you're seeing the early cancers or not, depending on how the antibody was raised. Have you looked into this? Or?
Yeah, no, that's a great comment and question. You're right. Like the glycosylation states will change for sure. One way to potentially overcome this is trying to target an area that isn't modified by the heparin sulfate, I should say. I think it's a high glycosylation. Sorry. Try to target some of the open areas. So the GC33 antibody developed by Shugai, for example, targets close to the membrane, so the just distal to where the heparin sulfate modifying regions happen to be. So it's ideally it would eliminate or at least minimize some of the issues we might run into if part of the binding region includes the heparin sulfate. But you're right; these are things that we need to flesh out, and particularly if we're doing phage display or the yeast display that I showed for the SCFB, if we're identifying some SCFBs that are specific to particular heparin sulfate moieties, then that may be altered, and then we'd lose that specificity. So you're right; it's definitely something that we're thinking about.
Is the Glypican-3 expressed in the normal tissues, or is it just more seeding the cancer?
So I don't know if I heard exactly, but I think you said whether it's expressed in the normal liver, right?
Correct.
Yeah. So it's not, and that's part of the reason why we're excited about it. The expression data that I showed from TCK and Tiger LSP, the control there is quote unquote normal liver, so tumor-matched tissue that's not HCC, for example. So it is a differential expression compared to, I don't want to say normal liver, because most of these are sick liver, right? But yeah.
Okay, Freddy, very nice talk. And I just want to, I think for Xin's questions, for cholangiocarcinoma, we have been shown years ago the mesothelin is one potential target, and we have made antibodies potentially can be useful to develop sensing targeting cholangiocarcinoma if we want to do imaging or therapy. And for GPC3, I think in general, in adult normal tissue, they are negative, especially on protein level and the cell surface level. We have done very comprehensive normal tissue array. And I think we just recently published a paper in gastroenterology, and Tim Gretton and I will actually developing a CAR T cells, and we actually showed all the tissue staining on the frozen tissue and also fixed tissues. And antibody we are going to use are not binding to the normal adult tissues. And so for the question for actually Freddy, so you are talking about from antibody to the peptide, and we normally, what kind of therapeutic window for affinity? Because for therapeutics, for example, the CAR T cells we are doing, and we don't really care much about affinity because there are a lot CAR on the T cells, and we really focus on the epitope. For antibody drug conjugate, we really need a very high affinity, sometimes very, very sub-nanomolar affinities. And what's the ideal affinity for antibody-rated isotope conjugate to get the best signal? I guess the too high is not good because you maybe have an antigen barrier there, and too low may lose the kind of specificities. What kind of the window there?
Yeah, no, that's an excellent question and point. We have to go for the Goldilocks version of this, right? So if we extrapolate from the data that exists for the neuroendocrine tumors, so if we're doing peptides, for example, which is a modified peptide, the octreotide, the affinity for that is in the order of tens of nanomolar for one of the serotonin receptors, SSTR2, and in the hundreds of nanomolar for the SSTR5, which is one of the other somatostatin receptors expressed in that tumor type. So that's still sufficient to see some effect, right? And we were able to see that overall survival. So we may not need an affinity that is really sub-nanomolar in order for us to see an effect. There's examples of that. In contrast, for PSMA, the ligands that are being developed for imaging are sub-nanomolar, right? And we get very good imaging. But what we're seeing is that there is a natural expression of the PSMA I mentioned earlier in the salivary glands. And because of this, if we do a therapeutic ligand that has the same affinity as the imaging, as in sub-nanomolar, we actually end up ablating and killing off a lot of the salivary glands, and patients can develop really bad xerostomia, so dry mouth. So actually degrading the affinity in that circumstance is able to better give you a therapeutic window where you still get some good accumulation in the tumor, but less so in the normal expressing salivary glands. So to some extent, it's going to depend on what we see in humans. The fact that the staining in normal human tissue suggests that the GPC3 expression is not very high. That suggests to us that we may be able to get away with a very, very high affinity ligand. What that number needs to be, I think, remains to be seen, though.
Okay, any other questions? If no questions, I don't know, Ting and Xin and Anu have any announcement?
I don't have anything about. I just want to thank Freddy. This is a very nice talk, and I agree with the team. Wonderful.
Thank you. Appreciate it. Thanks for the invitation.
Thank you, Freddy. Very nice talk. Yeah, always learn new things from your talk.
Great. All right. Thanks, guys. Take care. Have a good one.
Thank you.